Hello, my name is Pumika, and this lesson is going to be about the structure of nucleic acids. Now, this is only half the chapter. I'm going to include the second part in part 2, so please check that out as well. Let us begin. Okay, before going about purines, a simple definition of nucleic acid would say that successive nucleotides, they join to each other to give rise to long chains which are known as nucleic acids. Now we're going to study the basic concepts of nucleic acids, starting with purines. Now, we have heard this term many a times, but we usually get confused between purines and pyrimidines. So, a simple point to remember is that purines, these are double-ringed structures. As you can see here, we've got guanine and adenine, and one ring, two ring, one ring, two ring, and these kind of look similar to each other, so therefore we'll be calling these purines, and because they've got nitrogen in them, they're also termed as nitrogenous bases. So adenine and guanine, purine. Why? Because these are double ring structures. What are pyrimidines? As you can see, these are single ring structures. You can see only one rings in both the structures. This is the cytosine and this is thymine. Of course, they too have nitrogen in their structures. Therefore, these are also referred to as the nitrogenous bases. Okay, understanding the bonding. The basic bond that occurs in nucleic acids is termed as a phosphodiester bond. Now, how is a phosphodiester bond made? The simplest components, or the basic components of nucleic acids, are phosphate, a sugar, and a base. Now, for the phosphate, I have drawn here the triphosphate group. We've got 3 phosphate atoms here. Then we've got a pentose sugar. I have taken here deoxyribose. Why is this known as deoxyribose? Because the ribose sugar is a pentose sugar which has got OH here, but here we've removed that OH and we've only have H and H. So this is deoxyribose. And then we've got a base. I've taken cytosine for the convenience. Now, we also have some terms termed as nucleotide and nucleoside, which are pretty confusing as well. So, what is a nucleotide? Now, 1 plus 2 plus 3, that means this structure plus this structure plus this structure is termed as a nucleotide. When a phosphate group is attached to the pentose sugar and a pentose sugar is in turn attached to the base, then it gives rise to a nucleotide. Now, when I remove this phosphate group from all the 3 structures, only a sugar and a base remains. These 2 structures, when are bonded to each other, are termed as nucleosides. I hope the difference is clear. Now let's move on to the actual bonding. Okay, now I have a nucleotide here. As you can see, this is a nucleotide. We've got the phosphate group here. Only one phosphate, one P remains here, and the rest 2 are given out in the form of pyrophosphate. So only one P contributes to the bonding. So here we've got a phosphate, here we've got a pentose sugar, and here we've got the base. So we've got a nucleotide here. One nucleotide, when comes in contact with the other nucleotide, it forms a bond in such a manner. H2O is given out from here and here, if you can see, and as such it gives rise to a bond which is termed as a phosphodiester bond, this central part right here. So this phosphodiester bond is basically responsible for all the bonding that takes place in nucleic acids. Now, there's one thing that I skipped out here. Guanine, that means a purine, and pyrimidine, it has specific number of bonding. One guanine forms bond with a cytosine through hydrogen bonding. What is a hydrogen bonding? In which hydrogen acquires a partially positive charge because it is bonded to a very highly electronegative atom such as fluorine, oxygen, nitrogen. So here we've got a number of nitrogens. So these form hydrogen bonding with this. Only a guanine can bond with cytosine and only an adenine can bond with thymine. Now, it is important to know that guanine forms triple bonds with cytosine and adenine forms double hydrogen bonds with thymine. So, A double bond T, G triple bond C. Many a times in competitive examinations, certain questions are asked in which the most simplest structures of DNA are given. You are only to count the number of G and C, and whichever structure contains more number of G and C, that's the answer. That will be the most difficult to denature, to break down. Why? Because a triple bond is certainly difficult to break than a double bond. The bond length decreases, and as such it is pretty difficult to break it in comparison to the double bond. So I hope this lesson is clear. I did not want to include the structure of DNA into this because at a stretch it's very difficult to understand. I will be including in the structure of DNA in the second part, which is structure of nucleic acids part 2. So you might as well check that out. Thank you for watching my lesson.